Well, good afternoon, everyone, or good morning or good evening, depending on where you're coming in from. I just want to start out by saying that this RNA Collaborative is a shared event between University of Rochester and University of Michigan. My name is Eric Wagner. I'm a professor at the University of Rochester. I am the co-director of the RNA Center here. I think many of you know that this center was founded by Lynn McQuat 20 years ago, and she directs it today. I want to add one more thing that recently Rochester and State University of New York at Albany have a joint venture in New York State called the Center of Excellence in RNA Research and Therapeutics. And the goal of the CERT is to promote RNA biology in the workforce in New York State. It's actually a funded entity, and we have a modest budget now, but it's growing over time. And I use that as a segue to introduce our first speaker, Dr. Doug Anderson. Doug Anderson is an assistant professor here in the Cardiovascular Research Institute. But equally important, he founded a company called Stitcher, where he is the chief scientific officer. And I'm proud to say that the CERT has been able to help financially support that entrepreneur spirit. And we're going to hear more about that work today. So with that, I'm going to stop speaking and hand the floor over to Doug.
And happy to take any questions. Here's the PubMed ID for our recent publication, and if you have any questions as well.
Okay, I think that is all the time we have for the Rochester half. So I want to thank Doug. That was a great talk, and I'm going to hand things off to Nils Walter.
Thank you.
Thank you very much. Wonderful talk. It's great to be teaming up with the University of Rochester RNA Biology Center. My name is Nils Walter. I'm a professor of chemistry and the co-director with Mats Langman of the University of Michigan Center for RNA Biomedicine, which we founded in 2016, so a little more recent compared to Rochester's. In 2019, we received a significant internal grant that allowed us to hire 5 new faculty colleagues to augment our now 170 faculty membership. Michelle Hastings, who we hired into the pharmacology department in particular, is now leading, together with Peter Todd from neurology, a new initiative in RNA Therapeutics, for which we are currently fundraising. We collectively obviously founded the RNA Collaborative as a response to COVID-19 pandemic, and it is great to see that it's still going strong to this day, one of the few positive outcomes, I might say, of the pandemic. And it's my particular pleasure today to introduce 2 of our junior colleagues here from Michigan. The first is Grace McIntyre, who graduated from Marian University, Indianapolis in 2022 with double degrees in biology and public health. She then joined Dr. Analisa DiFeo's laboratory in pathology here, where she's a molecular and cellular pathology graduate student, as well as an NSF GIFP, and a Barbara Ann Robson ovarian cancer research fellow. And she's studying novel therapeutic avenues in ovarian cancer, where she's sharing with us her thesis work now on a high throughput identification of small molecule inhibitors. And second, after that, will be Adrien Chauvier, who grew up in French Guiana, in a smaller country in South America, and then moved to France with his French citizenship to get a bachelor's and master's degree at the University of Pau-Sabatier in Toulouse, and in microbiology and agrobiosciences. And then he moved to Sherbrooke, Quebec, Canada, another French-speaking place, to get another master's degree first in microbiology, and then a PhD with Daniel Lafontaine in molecular and cellular biology from the University of Sherbrooke. He joined my lab in 2017 as a postdoc and is now a research lab specialist, senior and lab manager in biology. So, but Grace is taking it off. Please start.
Okay. Thank you, Nils. I really appreciate that kind introduction. Today, I will be sharing with you all a high-level overview of my thesis project, which aims to identify small molecule inhibitors targeting Oncomere 181A for cancer therapy. So cancer therapy is challenged by the diversity of molecular signatures across cancer types. In a study of integrated genomic DNA or genomic data across 12 different cancer types from TCGA, recurrent regions of copy number changes can be mapped as shown here. Now, there are several regions that stand out as hotspots for copy number amplifications, and one of these regions includes chromosome 1, and specifically at 1q32, where more than 7% of cancer samples are amplified, we see the microRNA 181A is located. Now, if we take a look at about 5,500 patient samples and dichotomize them into two groups, which have either greater than or less than 2 copy number alterations in miR181A, we can see that miR181A copy number variation is associated with decreased overall survival in a pan-cancer setting. If we then dive deeper into various cancer lineage-specific cohorts, we can see that high expression of miR181A leads to reduced overall survival amongst patients with complete response, progressive disease, and stable disease in ovarian cancer. Now, if we look into other cancer lineages, we see that this remains true as well. Furthermore, we see reduced survival for patients with high expression of miR181A in breast cancer, colorectal cancer, gastric cancer, pancreatic cancer, and multiple myeloma. Now, taken together, we can see that miR181A truly is an oncogenic microRNA. Now, interestingly, these cancer lineages, which are driven by high miR181A activity, also have some of the highest numbers of estimated deaths among all cancer subtypes, suggesting that miR181A is a critical oncomere requiring further investigation. Now, mechanistically, miR181A dysregulation can enable cancer cells to acquire hallmark capabilities required by this neoplastic disease, including aberrant activation of several cellular pathways involved in tumorigenesis, including TGF-beta, STAT, Wnt, P10, AKT, and MAPK. Thus, regulation of miR181A is critical for maintaining homeostasis, and its dysregulation is concomitant with cancer pathogenesis. Now, our group and others have accumulated mounting evidence suggesting that microRNA 181A has a central role in driving cancer. In an ovarian cancer model, orthotopic miR181A overexpression drives metastasis. On the right here, we see that without miR181A overexpression, the tumors grow and develop but remain contained to the ovaries, leading to their immense size. However, upon miR181A overexpression, we see the migration of these tumors beyond the ovarian bursa and into the peritoneal cavity, indicating the true metastasis-driving power of this microRNA. Conversely, inhibition in vitro and in vivo in numerous mirroring cancer models has been shown to reduce cell proliferation, tumor growth, metastasis, ascites, and increase survival. In this model of a highly aggressive den-induced hepatocellular cancer model, we see that knockout of miR181A reduces tumor number and volume drastically. Thus, we believe that miR181A serves as an ideal therapeutic target in cancer. While no FDA-approved microRNA therapeutics exist, there have been several strides, with a total of 10 clinical trials having been conducted to target microRNAs in cancer. Recent studies have even identified potential small molecule modulators of miR181A through machine learning models and pilot screens. Using this information, my objective is to develop an unbiased workflow by which small molecule inhibitors can be screened, validated, and optimized to target miR181A. As a brief reminder, microRNAs are small non-coding RNAs that mediate post-transcriptional repression of mRNA by directly binding to the 3' UTR of mRNA, which contains a binding site complementary to a seed sequence within the microRNA itself, ultimately leading to the degradation of that mRNA of interest. To model miR181A activity, we generated and validated a functional biosensor containing 4 miR181A binding sites from its known targets. These binding sites are distal to an mCherry reporter. miR181A binding then mediates repression of mCherry, resulting in an inverse correlation of mCherry fluorescence and miR181A activity. So we stably express this construct in a high-grade serous cancer model, OVCAR8, and then we flow sorted for those cells most highly expressing miR181A, thus having low mCherry fluorescence. These miR-181A addicted cells were then used to complete a high-throughput screen of more than 54,000 drug-like compounds, each meeting Lipinski's rules. In collaboration with Dr. Aaron Robita from the Center for Chemical Genomics, we screened just under 55,000 compounds. We then used a ratio to select those compounds that both reduced cell viability and increased M-cherry fluorescence by greater than 3 standard deviations away from our negative control, resulting in about 900 compounds. We then screened those 900 compounds, searching for those that increased M-cherry fluorescence by greater than 20% at the 48-hour time point, resulting in about 355 compounds. We then validated these compounds across a dose-response curve to validate this reduction or this increase in M-cherry fluorescence and reduction in miR-181A activity, leaving us with 32 compounds. We then conducted structure-activity relationship analysis on 8 of our top hits, based on their potent inhibition of miR-181A activity and reduction of cell viability, leading us to an expanded hit compound selection of 49. After identifying our top compounds through an unbiased high-throughput screen, we wanted to know how many of them were predicted to be RNA binding. Our collaborator, Renju Jacobs, used a machine learning model comparing the compounds to known RNA binding compounds from the Robbins and Paynes datasets, comprised of more than 2,000 known RNA binding small molecules. We found that 94% of our top hits were predicted to be RNA binding with an average RNA binding score of 69%. From this set of 49 compounds, we were able to classify them amongst 3 structural groups. The remainder of today's data will be focused on our most promising class, Class X, and its 29 derivatives. In our high-throughput screen, we were able to measure the changes in M-cherry fluorescence at 4 doses as demonstrated by the red and purple here. Notably, increased fold change here is indicative of reduced miR-181A activity. Conversely, we were also able to measure changes in cell viability at 4 doses depicted here in green. Here, a lower fold change is indicative of reduced cell viability. We found that compound X and its derivatives demonstrated strong inhibition of miR-181A and reduced cell viability. Some compounds even inhibited miR-181A activity more effectively than our parent compound X at the 48-hour time point, including compounds X1 through X7. Correlation assessments of these compounds at the 48-hour time point showed R-squared values greater than 0.6 in linear regression analysis, suggesting that these compounds have a dose-dependent regulation of miR-181A activity. Particularly, compounds X1 and X6 showed significant linear correlations. To validate how this class inhibits miR-181A activity, we explored potential mechanisms by which these compounds could target miR-181A through different points in its biogenesis pathway. Upstream microRNA therapeutic approaches have previously targeted microRNA binding sites or interactions, including sites on dicer, drosha, microRNA stems and loops, or other microRNA binding proteins. So we began by measuring these compounds' effect on mature miR-181A expression. At an early time point of 6 hours, several compounds had reductions in miR-181A expression, including compounds X10, X7, X9, X71, and X12. We then looked upstream at primary miR-181A expression and found that some of these compounds inhibited both pri and mature miR-181A expression, suggesting that these compounds could potentially be acting on modulators of the primary miR-181A, and their effects are then sustained through maturation. We also see that compounds X10 and X7 have increased primary miR-181A expression despite reduced mature expression, indicating a positive feedback loop commonly seen when mature microRNAs are decreased. Next, we wanted to validate our high-throughput screen and see how changes in miR-181A expression correlate with potency in killing our cancer cells. We conducted a clonogenic assay and found that 93% of our compounds had an IC50 less than 4 micromolar. Interestingly, if we isolate those compounds that had greater than 20% knockdown in miR-181A, we find that an average IC50 for these compounds is more potent than those without miR-181A knockdown. After characterizing the potency of this class, we wanted to analyze cell death markers. At an early time point of 6 hours, we see that in addition to our parent compound X, compounds P33, P32, S71, X1, and X12 induce DNA damage and apoptosis. At the 24-hour time point, we were unable to run 5 of our compounds due to insufficient protein as a result of high cell death. Therefore, these lanes were left empty as placeholders. But with the compounds that we could run, we found that P34 and S05 now had elevated cell death markers at 24 hours. Overall, this suggests that this class does induce DNA damage and apoptosis. Next, we were interested in understanding how this class of compounds impacts the promoter activity of miR-181A and its known downstream targets. We tested the promoter regions surrounding the miR-181A1B1 transcription start sites by using a luciferase reporter vector. Interestingly, nearly all of the compounds had either no effect or upregulated miR-181A promoter activity, suggesting a positive feedback regulatory mechanism activated by downstream miR-181A inhibition. In contrast, compound X10 significantly downregulated miR-181A inhibitor and miR-181A transcriptional repressor OTX-015, which suggests that this compound could be transcriptionally regulating miR-181A. Lastly, in collaboration with Dr. Sriram Chandrasekaran, we were able to conduct a machine learning analysis where the structure of the 29 compounds from this class that we were interested in was compared with the structures of more than 80,000 proteins. This analysis enabled us to predict potential compound-protein interactions and their predicted dissociation constants. In this analysis, we identified 42 proteins with a predicted log KD greater than 6.5 and more than 85% of our 29 compounds. This group included 9 keratin-associated proteins, 4 zinc finger proteins, and 6 endosome organization proteins, suggesting that this class could be affecting these particular processes. In an analysis of 5 compounds from this group, we sought to identify common predicted protein interactions. We found that 4 out of 5 compounds were predicted to bind to WDR5, a component of an epigenetic regulator, and H3BTT4. In conclusion, we have preliminarily screened 16 compounds by looking at their effects on miR-181A expression, potency, and on-target profile. Our ideal compound would reduce miR-181A expression, potently induce cell death, and increase the protein expression of the known miR-181A targets. While our parent compound meets nearly all of these characteristics, we would have been able to identify some other compounds like compound X1, 9, 10, 7, and 12, which carry more of these desirable traits. Now that we have this set of desirable compounds, future studies will begin to assess the compound interactions with miR-181A and its predicted proteins. Upon completion of these studies, we will identify our hit compounds and then complete other studies to assess its broader effects on small RNA and global mRNA and protein expression. And finally, we hope to complete this work with toxicity and in vivo studies. With that, I'd like to thank my incredibly supportive lab for all of their work and my undergraduates who have worked really hard on this project with me. I'd also like to thank the MCP graduate program and the CCG here at U of M, and I'll take any questions.
Thank you, Grace. Very, very nice talk. We already have a question here, which actually I might have asked as well. And I think you anticipated already a little bit. How do your compounds affect the level of other microRNAs? Could it be essentially a broad transcription inhibition that you see?
Yeah, that's a great question. So we've been able to, because we're working with such a broad group of compounds at this point, we haven't been able to run small RNA-seq yet to assess the effects on like total global microRNA expression levels, but we have looked at a couple other microRNAs, and we have seen that about half of our compounds may reduce additional microRNAs, and some of them have no effect on microRNAs. So we're hoping to potentially use that to identify what structures are going to be of greatest interest to us.
Thank you. And as the host, maybe I can ask one final question then. So you know what the structure of the molecules is. Unfortunately, you couldn't show us, but they are RNA binding. What's the prospect of it binding an RNA relatively specifically, either sequence specifically or structure specifically?
Yeah, that's a great question. It's not something that we've been able to look at yet. We have a couple RNA binding assays that we're hoping to do in the next couple of weeks, so hopefully I'll have a better answer for that.
All right. So with that, thank you very much, Grace. Again, wonderful talk. And to stay in time, we now switch over to Adrien, who will tell us about real-time monitoring of ligand recognition by Riboswitch during transcription. Adrien, take it away.
Right. Hello, everyone. I'm very happy to be here and to present some of the work that we are doing in the Nils Walter lab. So I believe I will not hurt anybody's feeling when I say that RNA are master regulator of gene expression. And this is particularly true in bacteria that employ numerous bacterial non-coding RNA to regulate and fine-tune gene expression as a function of the environmental cues. This is particularly true and present at numerous processes where non-coding RNA can regulate their own transcription and RNA stability or even translation of the subsequent gene. In the Walter lab, we focus on a specific bacterial non-coding RNA that act in cis that's called riboswitches. Riboswitches are master of gene regulation, and they can bind a specific metabolite or an ion as that we term a ligand through an aptamer domain. Then, upon binding of the ligand, there is a change of conformation of the subsequent expression platform that will allow regulation of gene expression, either at the level of the transcription, controlling transcription termination, or at the level of translation, preventing or regulating the accessibility of the Schalder-Garneau and the start codon to regulate initiation of translation. All right. What is intriguing for me in terms of riboswitches is that in some bacteria, they can control up to 4% of gene, and most of the time, those genes are essential for the bacterial fitness and survival. Therefore, they are promising target for the design of antibiotic, as we know for the emergence of antibiotic resistance. They are promising target to elevate that. So for today's story, I will present the work we are doing on the Bacillus subtilis BLIQS T-box riboswitch. This riboswitch controls the expression of amino acid synthetase upon binding of a tRNA. So as you can see on this in vitro transcription assay, as we increase the amount of tRNA or uncharged tRNA, we increase the formation of a full-length product, showing that this riboswitch is acting as an on switch, promoting expression of gene expression. I found this type of riboswitch and all the T-box riboswitch very intriguing because they go beyond the simple distinction between aptamer and expression platform. In other words, while the riboswitch is able to sense a specific tRNA through the anticodon and the specifier loop in the so-called aptamer region, it can also discriminate the tRNA based on its charge. And this is localized into the expression platform, where in the absence of the amino acid, of the charged amino acid, the 3' end of the tRNA will interact with the so-called T-box motif, promoting the formation of the anti-terminator. However, if there is no tRNA, or if there is an amino acid on the 3' end, there is a steric hindrance, and the formation of the terminator is promoted. So in recent years, there are numerous structural insights about this riboswitch in complex with the tRNA through X-ray crystallography or even cryo-EM that pinpoint some specific interaction and help us to understand how this riboswitch works in combination with the cognate ligands. However, very less is known how this riboswitch senses specific ligand, and especially during the transcription. So to fill this gap, we engineered a fluorescent version of the tRNA, placing a fluorophore on the elbow of the tRNA to allow for subsequent single molecule analysis. And as you can see on the same in vitro transcription assay, the presence of the fluorophore on the tRNA does not significantly affect the genetic response induced by tRNA binding to the T-box riboswitch, allowing us to use this fluorescent version for subsequent single molecule analysis. All right, so to start my investigation, I wanted to know how the tRNA is sensed during transcription, and more specifically at key transcription points. So what I did is that I immobilized a pause elongation complex, meaning that I stalled the polymerase at the position of known transcriptional pause sites that has been identified before in vitro and in vivo. And I can fluorescently label this pause elongation complex, immobilize this on the microscope slides, and upon direct excitation of Cy3 and Cy5 fluorophore, I can visualize tRNA binding in real time at the single molecule level. And this is what you can see here on different pause elongation complex that I used in this study, encompassing either only stem 1 or the so-called aptamer domain, and then moving through the expression platform at the position 178 that allow the exposure of the T-box motif, and through 207 where the anti-terminator is completely folded. So as you can see on those representative traces, tRNA binding is very transient and for a short period of time. And this is better visualized through the distribution of the binding events, where I sort them by their tRNA lifetime. In black, they are short or less than 5 seconds, and in red, longer binding events that we assimilate as capture of the tRNA during transcription. As you can see here, upon the distribution of the tRNA binding to the T-box riboswitch, is that as we increase the length of the transcript, more and more binding events and especially more capture of the tRNA is observed, especially at the position 207 that allow the formation of the anti-terminator. To evaluate how the RNA polymerase impact the tRNA binding kinetics, I transcribe the same or relatively same RNA lengths, but in the absence of the RNA polymerase. And this is what we call because they are released from the RNA polymerase. And we can extract different information from this study. First is that when you have just the stem 1 or the aptamer domain, very short binding events are observed compared to the same complex in the presence of the RNA polymerase, suggesting that the presence of the polymerase promotes binding and capture of the tRNA. And then, as we increase the length of the expression platform, we observe that the most efficient transcript is shifted backward compared to the pause elongation complex. However, when you inspect the secondary structure of the transcript that is accessible for binding the tRNA, we observe that actually 207, the presence of the polymerase at 207, prevent the formation of the terminator that will impact the switching mechanism between anti-terminator and terminator, showing that actually the presence of the RNA polymerase is the determining factor for allowing tRNA capture and binding during transcription. All right. So to better understand the critical determinant for binding of the tRNA and capture during transcription, I isolate my PEC-207, the most efficient for tRNA binding and capture, and I introduce mutation into the expression platform that are predicted to affect tRNA binding and capture. I use the M-box, or mutating the 3' recognition site of the tRNA, and I observe very short and transient binding events. I also introduce mutation in the anti-terminator to test the stability, the effect of the stability of the anti-terminator into tRNA binding. Again, if we look at the distribution of the tRNA binding events in the wild-type construct, we observe a lot of short, but also a significant amount of capture tRNA that is completely abolished when we mutate the 3' recognition site, as expected from the literature. However, we were very surprised to see that when we mutate the anti-terminator, the MAT construct show less and significant no capture of the tRNA. And that suggests that actually folding of the anti-terminator will help for exposing the T-box motif for subsequent capture of the tRNA. And this is confirmed when I re-established the anti-terminator structure through a compensatory mutation. The MAT-CP construct shows significantly more binding events and more captured tRNA during transcription, even more than the wild-type construct, actually. And this is further supported when we look at the kinetic analysis of tRNA binding during at post-CO7, namely the unbound time or the time between binding events. We found that actually the anti-terminator is very important to recruit the tRNA. And when we look at the tRNA bound time, we observe that the integrity of the anti-terminator is necessary to allow efficient tRNA capture during transcription. This analysis allows us to pinpoint the specific determinant for tRNA binding and capture during transcription. But I said during transcription, but so far I'm looking only at snapshot, just key points during transcription. And we were very interested to know how tRNA binding kinetics occurs during active transcription. And this is why I developed an in vitro single molecule fluorescent platform to monitor transcription in real time. In this particular setup, the short pause elongation complex right after initiation is immobilized on the microscope slide using the transcript that has been labeled with the biotin. And I can visualize my molecule on the slides because I put a side tree on the 3' end of the DNA template. And that becomes very important because when we inject in real time the NTP substrate and the tRNA that is labeled with Cy5, we can monitor transcription in real time until the polymerase reaches the end of the DNA template. And this is marked by the occurrence of a PIF signal or protein-induced fluorescence enhancement when the protein bump or come into close contact with the Cy3 fluorophore. And this is what you can see on these representative traces. We know when we start the transcription and we know when the transcription stops. So we can delimit the transcription process through a co-transcriptional window and observe tRNA binding during active transcription and even after the transcription because the transcript is still attached to the microscope slides. So I performed this experiment with just the stem1 or the aptamer domain, and the data are better visualized through those rastergrams. In this representation, the molecules are actually synchronized based on their transcription time. So a molecule that is visualized at the bottom here is a fast molecule that takes a short time to transcribe. When the molecule is observed at the top here, that means that the molecule has taken a longer time to transcribe. And we can visualize and sort the binding events during and after the transcription. All right, you can notice that as expected before, binding of the tRNA are relatively short and sparse during transcription and relatively rare when you use only the aptamer domain. However, when I transcribe a longer version, the full-length Riboswitch, right before the decision point, you see more and more binding events during transcription, also after transcription. And then this is when we can visualize the tRNA capture during transcription. And as expected, as an internal control that the experiment is working as expected, the transcription time is longer. That's not only explain the number of binding events, but also that we have a sparse transcription time, probably due to the presence of transcriptional pausing during transcription. So this experiment is a source of so many information. I don't have the time to go over all of this, but we can survey the tRNA binding during transcription. And I use numerous mutation into the riboswitch to understand how the riboswitch sense its ligand. So I was surprised at first that when I mutate the T-box motif, I did not see a significant effect during transcription. However, when I use the mutant of the aptamer domain, a known dual T-loop motif, the DTM motif, I saw very less binding events. That's relatively expected because here you affect the initial sensing of the tRNA. And then we can focus on the first binding events. What happened when the T-box first encountered the tRNA? And this is when the information become more clear for me in terms of how it sends the ligand, because the M-box, yes, saw less binding events, but it's more pronounced in the delta DTM. So if you affect the first recognition site, you affect the early tRNA binding, and we propose a model where the tRNA needs to be first sensed, and then that prepare the tRNA for a subsequent capture. To understand this in more detail, I went into a more simplified system where I isolate stem 1, and I observe the kinetic of tRNA binding during in this construct, and I observe that is bind tRNA using two kinetic regime. One is very slow and unlikely to occur in vivo. However, we observe a very fast recruitment of the tRNA in the wild type construct that is completely lost when we mutate the DTM construct, allowing us to postulate a folding mechanism that happened during transcription, where the folding of the DTM, the top part of the stem 1, allow for a better recruitment and subsequent capture of the tRNA. To mimic what would occur in terms of in vivo condition, we can play with the condition on the microscope slides. And I first wanted to investigate how the transcription rate, the rate of transcription will affect tRNA sensing. So I increased the rate of transcription by increasing the amount of NTP used during the transcription. As you can see that the transcription rate now is shorter and coming much faster, and obviously you see less binding events of the tRNA. As an internal control and to make sure that I don't have an unforeseen effect of the NTP added, I use the RNA polymerase mutant that is slow prone. And you can see that this time the transcription time is slower, as expected, using this mutant version of the RNA polymerase. I also wanted to understand better how transcriptional pausing will affect the kinetic window for tRNA binding. So I use truncated version in those conditions at the known pause 178 and 207 that I used before. And I was very surprised to see very few, very less binding events, suggesting a very narrow kinetic window for tRNA sensing and tRNA binding during transcription. This is better summarized here on the distribution of the binding events. Basically, the rate of transcription has no significant effect on the kinetic of tRNA binding. It has more of an effect into the amount of how much tRNA can be sensed by the tRNA that support a kinetic window for tRNA sensing. But how to explain that it occurs during transcription in the fast kinetic product is that we propose that transcriptional pausing provides a kinetic window, more time to sense the tRNA. And actually, this is when we look at the post-transcriptional window that we have more information about this. As you can see, the rate of transcription is not affecting the distribution of the long and short binding events for a full-length construct. However, when you, as much as you increase the length of the tRNA and allowing more thermodynamic stability of the anti-terminator terminator construct, you see more and more of the captured tRNA. This is better visualized here through the kinetic analysis, where as much you increase the length of the T-box riboswitch, you see more and more reaching a maximum of 30% of long-lived T-box tRNA capture during transcription. All right. Altogether, Among other experiments that we performed, we present a co-transcriptional model for tRNA sensing during transcription that is mediated by a hierarchical folding of the T-box riboswitch. This starts from stem 1 synthesis that allow for the tRNA to be fished, to be selected from the total pool of tRNA and select the specific lead tRNA in that case. And we propose that the pause at 138, the first pause right after the synthesis of stem 1, allow more time for the DTM or the top part of stem 1 to fold and pre-capture the tRNA. All right. And then you have two scenarios that can occur. Is that the bacteria is under stress. Everything is shut down. The transcription is very slow. That will allow more time for subsequent capture of the tRNA and more time for the anti-terminator to fold at the pause 207, providing a last checkpoint for tRNA capture to promote transcription read-through at the decision point, which is the termination point. However, when the bacteria is thriving, very happy, all the resources are available, transcription is very fast, giving less time for capturing the tRNA, and the presence of the charged tRNA that account for the majority of the tRNA present in the cell under good condition, if I can say, prevent the inefficient tRNA capture, and then the transcription continue to terminate and to prevent the use of resources that will not be used by the bacteria to survive in thriving condition. I will end obviously to thank you all for your attention, thanking my advisor and friend Nils Walter for his support and constant trust, my amazing team. We work all together, and thank you all for your support and to bear with me when my excitation is too much, let's say. Collaborators, obviously, at Rice University that provide us for the fluorescent version and design of the tRNA. Dr. Javier Cabello Villegas that initiated this project as first, and obviously all the funding agency that allow us to do what we love, science, and ask specific biological questions. Thank you.
Thank you, Adrien. Very, very nice talk. Good overview. I know we are running a little late, but there is one time for one quick question. That is on, I mean, first of all, nice study, it says, by Mark Foster, and then shouldn't tRNA binding modulate termination? And if that is observed, is it avoided by, or is it not observed, and is it avoided by modifying the template? What are the effects on tRNA binding?
All right, so thank you, and interesting question, obviously. So to be able to survey, at least for the real-time transcription, I stopped the DNA template right before the termination, so that I can keep my template on the slide and not lose my molecule, because this is what is very difficult with those kind of study. You want to keep your signal because you want to see the PIFE event. If I place the fluorophore after the termination site, I will lose the fluorescent signal because there is dissociation. However, that's allow me to introduce, maybe if I have the time, another method that we design is this one. We are actually able in the Walter lab to survey transcription termination regulation by putting two fluorophores on the DNA template. So this time we put a fluorophore before the poly-U sequence and after the poly-U sequence, and we observe what we call a double PIFE in case we have transcription read-through, and only a single PIFE event when we have transcription termination. So I did not have the time to present this, but this is the kind of tools that we can develop in this study.
Thanks, Adrien. And it just is up to me then to close this off and just let everyone know that the next meeting is on May 14th when the Institute of Molecular Biology and RNA, the RNA Research Center in Poznan will be presenting. More information will be on the webpage for the collaborative. So thank you very much all for your attention and great talks all around. And so see you around.